Habits begin with a QAN external or internal signal that activates sensory processing networks. Visual information travels from the retina to the primary visual cortex within milliseconds. The prefrontal cortex interprets this signal, assigning meaning and context. At this early stage, behavior remains under conscious evaluation. The brain is still deciding what action to take. With repetition, control begins to shift. Neural signals increasingly engage the basal ganglia, particularly the striatum. This region integrates context, action, and expected outcome. As behaviors are repeated, synaptic pathways become more efficient. Decision-making gradually requires less conscious oversight. Reward strengthens the loop. Dopaminergic neurons in the midbrain release dopamine into the nucleus accumbens. This signal encodes reward prediction rather than pleasure alone. When outcomes meet or exceed expectation, synaptic reinforcement increases. The brain begins associating the cue directly with anticipated reward. As repetition continues, neural processing becomes streamlined. Activity in the prefrontal cortex decreases during habitual behavior. The basal ganglia now trigger motor outputs directly. The sequence of actions is executed as a single behavioral unit. What began as deliberate choice is now automatic routine. Eventually, the brain compresses the entire behavioral sequence. The cue alone activates the full neural routine. Dopamine release shifts earlier, occurring at anticipation rather than completion. This anticipatory spike strengthens the loop even further. The habit is now neurologically encoded. When reinforcement becomes excessive, habits can dominate behavior. Reduced prefrontal inhibition limits conscious regulation. The basal ganglia loop activates rapidly and repeatedly. What began as adaptive learning can evolve into compulsive action. Understanding this circuitry is central to treating behavioral addictions and maladaptive routines.